grade 12s, how are you all doing? Welcome to Life Science, and today we're going to have a look at meiosis and mitosis. Right, so as we know, first thing when it comes to life science, we always look at specific things. We're going to have a look today in the beginning at your terminology, because once you know your terminology, you A for away. Okay, and in this section, I've pulled up a couple just so that you get used to it. Make sure that you remember how to do these in a test and use these words all the time. Okay, let's have a look at the first one. What I'm going to do is I'm actually going to give you the words. I'm going to give you a few minutes to try and give me some feedback first. The first one is meiosis, which you've done before, right? So that should be quite easy. Then you get, mitosis. Then you get meiosis. Okay, meiosis is a new one. Then I want you to have a look at gametogenesis. Another one is genetic variation. And then you've got haploid and diploid. Now, out of those, I know that you've seen haploid and diploid before. Okay, I know that you've seen mitosis before. So there's 3 new ones. Hopefully, you can get it right. And what I'm going to give you is 2 minutes. Let's see if you can get this right. Ready, go. Okay, so now let's have a look. You've got these words going. You've already started using those brains. This is your warm-up, right? Because I need to teach you new things. So let's have a look how good you were at the warm-up. The first one, mitosis. Now I'm going to change color pens here. If we have a look at mitosis, it's a type of cell division that results in 2 daughter cells, each having the same number of chromosomes as the parent's nucleus. And so far, these words you're understanding, right? And typical in ordinary cells, so your muscles, your skin, all that type of thing, and it's also for growth. Now, before I carry on with that, one of the things here is it says 2 daughter cells, okay? One thing you must also remember that with mitosis, they are identical, like 100% the same. There's no difference, okay? And that's one of the key things about mitosis. There's no difference, okay? Let's have a look at the next one. The next one we're having a look at is the new concept that I'm going to show you today, okay? It's meiosis. Now, they look similar, they sound similar, but they aren't. Okay, let's have a look at the first one. The process of reduction division. Reduction division, okay? Reduction division, it's a type of cell division that results in 4 daughter cells, you underline that, 4 daughter cells with half the number of chromosomes, which is very important, of the parent cell. Okay, so if you have a look at it, one of the things that's missing out of there as well is they are all unidentical. Okay, don't forget, they don't look the same. In mitosis, we ended up with 2 cells, and both those cells look identical to each other, and they look 100% to the parent cell at the top, right? With the meiosis, what's happening is you get the parent cell, and the parents make 4 cells, and every single one of them is different. Okay? That's the difference between the two. Now, where we use them is where we're going up later on. Okay, the next word that we're having a look at here is gametogenesis. Okay, now genesis is to make, right? Okay, so there's the genesis, and then you get the gameto. Okay, and that part there means to make gametes. Now, can you remember what gametes are? That's your sex cells, right? So your egg and your sperm, for instance, right? Or the egg and the pollen grain. Well, not the pollen grain, the gametes inside the pollen grain, right? So all of those things, this is where this comes from. So if you have a look at the process in which cells undergo meiosis to form gametes. Now, hold on. Now I've used meiosis in the sentence, okay? And remember, meiosis is reduction division. So we're going smaller. Don't worry, it's not that difficult. I promise you, I'll explain it in big detail, okay? And the next one, genetic variation. Okay, your genetic variation is changing in the genetic code. So if we look at the variation in the gene sequence of a genome, and those are big words, yes, but it's like changing in the gene is like if you have brown hair and your mom has, like you have brown hair, your dad has blonde hair, and your mom has blonde hair, and all of a sudden your hair is brown, something's wrong. It's not. It's a change in the genetic code. There's a reason for it. Okay, it's one thing that you got to remember. And yes, I use blonde. When you get there, you'll understand that you can't use blonde. But all of this is very, very important. Okay, and the next one, these are the words that you do remember. It's very simple. Haploid, right? The nucleus that contains a single set of unpaired chromosomes. Okay, and then of course, diploid it consists of 2 complete sets of chromosomes. Haploid can be represented as N, and diploid can be represented as 2N. I know you haven't forgotten that. Those are easy. That's like grade 9 work or grade 11 work. These are simple terms so far. Right. Okay. Hopefully you got it, and we're all happy. Let's look at key concepts. Key concepts, that's the things that we're looking at mostly in this section. First one, the purpose of mitosis. Come on. The purpose of mitosis is very simple. You're having a look at stupid things like when you cut yourself or you grade yourself, you have to make new skin. So we're talking about replacement, okay, repair, and of course, asexual reproduction and growth. I mean, let's just go with the order that we've got here. Growth, okay, if it wants to write today, okay, I look at growth. You started off as a single cell in your mommy's womb. Right, and what happens? You got a lot more cells and you became who you are today. That's growth. Those cells came from mitosis. Repair, as I said, you've scraped yourself and now they've got to repair that. We look at replacement. Cells get old, right? And if they get old, they've got to be replaced. It always happens. Then asexual reproduction. So if an animal like an amoeba, okay, goes through mitosis to create another amoeba cell, okay, that's reproduction. It's quite cool. Next one. The purpose of meiosis is the reduction, division, resulting in gametes, as I said to you, making of sex cells. So in other words, your sperms and your eggs. Right. The next one, the importance of meiosis, okay, the production of gametes, which are haploid cells from diploid cells, sorry, from diploid cells to haploid cells, right? And the genetic variation in them is one of the most important things. The variation is so important, otherwise we will all look the same, okay? And then the last one that we're going to have a look at, the consequence of abnormal meiosis. Okay, abnormal meiosis, which is your Down syndrome child. So in other words, where something went wrong in that splitting. Okay, so that's nice key concepts, and you guys will understand that. I promise you, it's not as difficult as it looks. Right, okay, let's go for it. We're going to have a look at meiosis. Now, you remember mitosis, you had one cell, I'm just going to write it here, you have one cell, and it splits down into two cells. Okay, and that was the whole process, one process. Now we're having a look at mitosis, meiosis. And meiosis is divided into two sections. And there you have them, all one section. And you remember those names? Sure you do. Interphase, prophase, metaphase, anaphase, telophase. Okay? There's a phase missing here. Well, here it's not. But there's a phase that actually doesn't work with us. Okay? And if we have a look at it, they've got an extra part on this phase. Here we go. We've got these Roman numerals. Can you see it? And they're very important. Because if I'm talking about meiosis and you don't say prophase 1, okay, if you don't say prophase 1, the first thing I think of is mitosis. You need to make sure that you put that exact Roman numeral with it. And there's one for process 1 and there's one for process 2. Don't forget it. Right. So if I take these guys, if we have a look at it, if I take these guys here as you see them and I start separating them, I'm going to show you how each thing works. Okay, let's start with the first one. Okay, the process of meiosis. There are two parts, as I said, namely meiosis 1 and meiosis 2. The phases in meiosis are, let's have a look at it. What you don't forget is ip-mat. It's ip-mat. Interphase, prophase, metaphase, anaphase, telophase. Right? It's the best way to remember it. Okay? Now we have here interphase. Now, you don't actually get an interphase in meiosis. Okay? That interphase is normally said in mitosis where you have between phases. Okay? And yes, you're going to get the replication of chromosomes, which is what we want in this, the same thing. Okay? But they start in actual fact, you get a G1 stage, then an S1, and then a G2 stage. And in the G2, this piece here, so in the G2, which you don't really need to know, but in the G2 is where you get the replication of the DNA. Okay, so in the interphase, in this case, you get the replication of DNA. Simple, easy. Okay, then we get to the prophase. There it is. Prophase is one of the most important phases. This is where everything starts coming to life. Okay, it comes to life. If you have a look at it, you've got a blue and you've got a red. See, there's the blue and the red. Okay, the two chromosomes. Now, they are homologous pairs. Homologous, homo meaning the same. Okay, and the logous pairs, it's because of what you find on those DNA sites. Okay, now, you get two chromosomes, and I'm going to use different colors here quickly. Okay, and you're going to have the one, and you're going to have another color. Well, it actually does that. Okay, well, in actual fact, I'm trying to make it as easy as possible so that we don't understand, that you do understand it perfectly. So you get the one like that, and you get a red one like that. Okay, and they join here in the middle. Okay, now what happens when it comes to meiosis? Each one of those duplicates. Okay, so we started off with a purple. Okay, and the purple becomes two of them. Okay, and they join together. And then you've got the red ones, and they join together instead of the purple and the red joining together, right? So they double. Can you see how the chromatids double? Okay, and they go and they mix. This is still homologous pairs. Okay, that's a pair of chromosomes. It's homologous, right? Now, what they do is they go sit next to each other. They grab hold of each other. And listen to how I said it. They grab hold of each other, okay? And they start doing things which is quite cool because they do this, for instance. There's the one. You'll see I'm doing that, okay? And the purple one comes like this. Look at this. Have you seen how they've crossed over? See how they're crossing their arms and their legs? So if you stand like this and you cross arms and legs, it's called crossing over. That thing is called crossing over. It's very important, okay? And the place where they cross over, I'm going to take a black pen here, and where they cross over, this place here is chiasma, okay? But you need to know crossing over. And the reason why they cross over, it's very important, is that later on, you're going to end off with something that looks like that, and it's got this small piece there, and it comes down there, and you're going to have a bit of that colors there and a bit of that colors there. See how it's changed? And then you have this one, it's a small piece, this one a big piece, and you're going to have the red over there and the red over there. Okay, now, the importance of this, if you have a look at it, the purple and the red. If I look at this purple piece here, this purple piece, let's say that comes from your dad. That's the DNA that you got from your dad, right? And then this one is the DNA that you got from your mom, okay? When they duplicate like this, and you'll see that when they get to this stage here, that you have some of your mom, which is over there, all the squiggly ones here, that's all your mom, and then on that chromosome you also have some of your dad. Can you understand? Okay, so this is called crossing over. It's very, very important. Okay, and this whole thing here, without this, we have a problem. Okay, now I'm going to keep going. I want you to just keep thinking about this. I'll give you a break in a few minutes. Trust me. Okay, but this is very important. Okay, now that happens in prophase 1. In metaphase 1, okay, remember metaphase 1, they go line up on the equator. As you can see over here, they line up on the equator. Okay, and as they lined up on the equator, there they are. Okay, they lined up on the equator. Oh, that was quite cool. I don't know how we did that, right? But as they lined up on the equator, remember you get your centrioles on each side, and they create these spindle fibers. Can you remember the spindle fibers? Okay, and they're going to sit over there and join the centromere that goes between the chromatids. But what I want you to understand is these guys that are lining up on the equator, when we went to mitosis, they lined up one next to each other and they all looked identical, and each one was pulled to their side, so whatever DNA was here is the same as the DNA here, okay? Now, when it comes to this one, they line up randomly. In other words, it doesn't matter how they line up, okay? As long as whatever, if we're looking at shoe color, shoe's going to go up there and shoe's going to go over there, and I've used shoe color because it's not a human, okay? So it separates. As long as each trait goes to different sides, it doesn't matter if it's a trait from your mom or the dad. Okay? And that's where they line up. They line up in different orders, and that's what happens in metaphase 1. Okay? Then you've got anaphase 1. They get pulled to the poles, as you can see. So as they get pulled, you're going to have the one that does that over there, and you're going to have the one that does that over there. I'm just going to do it like this. They're going to get pulled to the poles. Okay? That one goes that side, and that one comes this side. Okay? And if there was another one over here, it would do that, and this one would be pulled there. If there was another one over there, this one would be pulled there. Okay, can you see how they're being pulled aside, which is normal? You've seen this, okay? And then you get telophase, where you get cytokinesis, or the splitting of the cytoplasm. Can you see the splitting of the cytoplasm? Okay, now remember, if I took these two cells, remember I had one like that, and the other side was a little piece, okay, with a bit of the purple, and you had the other side, with a bit of the purple over here, the big side there with the purple, little piece of purple. Can you remember that original one that we started with? Okay. Now, and you've got to understand this, if I put this in a ring, there it is, 1, 2, each one goes into a cell. Does this one look identical to this one? It doesn't. Look here. That much of the purple is here. Look how much purple is here. Can you see it? And look how much red is here, and look how much red is here. Can you understand? So is this cell identical to this cell? No, it's not. Okay, so they're unidentical, which is the most important thing of meiosis 1. Right, okay, so now I can see the smoke coming out of your ears. I really can, and I know I've kept you quite busy, okay? But this is what I want. Go have a break. This is a quick understanding for it, and when you get back, we'll carry on. Welcome back. I can see you guys are looking a little bit better. It's not that bad. I promise you it's not that bad, okay? At the end of the last section, we ended off with 2 unidentical daughter cells. Right, now remember, we started off with this. We had this interphase that actually doesn't exist. It was, and then prophase, metaphase, anaphase, and telophase. That is your mitosis. To make it sure that it's meiosis, I need to put the ones in. Okay, it's meiosis 1. There they are. Right, meiosis 1, and that tells me that it's process 1 of meiosis. Right, and remember, interphase doesn't actually exist. It's just where we doubled the chromosomes. Okay, now we get to meiosis 2. This stage is this is similar to mitosis. This is the easy part. Okay, now let's have a look at it. There's the whole process. Okay, And what I have done is I've just left it like this. It makes it quite easy. Okay, let's have a look at it. Firstly, your interface. There is no such thing, and I left it there for a reason, because people always make that mistake. I left it on the slide, especially to show you that this, oh, sorry, wait, hold on. I'm going mad. I'm still on Prophase 1 here. Okay, that is never a good thing. So let me just go back. You can see I'm going mad here today. There we go. This thing, interface, if I take it from there, this is still in process 1. There it is, Prophase 1, Metaphase 1. Now you see how they're all working together. Anaphase 1 and telophase 1 with the cytokinesis. Remember that. Okay, now, before I go on to number 2, okay, I want you to understand that these stages are not like built stages. They link into each other. So if you want to see it, it's not like a hand in a hand. It's the hands mix one in front of the other. So they almost link. Can you understand what I'm doing? Okay, so don't think of it as a proper, this is what you will see, and this is what you will see. You get an early prophase and a late prophase 1, and you get an early metaphase and a late metaphase. Can you understand what I'm saying? They link into each other. So it's not just a set phase, right? Okay, as long as you remember that, because people forget about that. Let's go to the main things. Before we go into Metaphase 2, let's do some questions. Let's see how good you actually are, if you've been listening to me. Okay, here we go. Questions that I want. Briefly discuss what happens during Prophase 1. Okay. Describe the purpose of mitosis. Okay. Next one. How many times does the cell divide during meiosis? And the last one. What kind of cells are produced at the end of meiosis? Okay. Now, those 4 questions, around about 2 minutes. I'll see you after 2 minutes. Well, if I'm having a look at your faces, which I can see them, so don't think I can't, right? I don't think you're that confused. I think that you've hit the nail on the head. Let's see how good you actually are, right? Let's have a look at it. The first one, briefly discuss, okay? Was briefly discuss what happens during Prophase 1. Here we go. During Prophase 1, homologous chromosomes, remember I told you about homologous chromosome pairs, exchange DNA in a process called crossing over. That is the most important part of Prophase 1. Okay? Next one, describe the purpose of meiosis. Remember, I said it's reduction division. In other words, it's going to go smaller, and it makes, in gametes, it's reduction division, and it makes gametes. And the process is called gametogenesis. There will be random arrangements of the chromosomes. In other words, remember, they swap places. It doesn't matter how they line up, as long as they line up and they go to the right places, right? And then the last one, well, the second last one is, how many times does the cell divide during meiosis? 2, remember, that's why we have metaphase 1, for instance. Remember, okay? And the last one, what kind of cells are produced at the end of meiosis? Haploid daughter cells, or N cells, right? So that was easy. I know you got those answers. And if you didn't get those answers, remember, you need to concentrate and make sure that you understand what's going on. And your teacher is there, or you can go and find out. I promise you it's easy. It's not as difficult as you think. Okay, now let's go. Now I want to get on to metaphase, to meiosis 2. You can see I want to get into it because meiosis 2 is the quick and easy one. This is the fun one. Okay, this is the same as mitosis. Okay, and you represent it as like, remember, you got IPMAP. IPMAT. Remember that. That was in metaphase, in meiosis 1. With this phase, you don't, and I'm doing it like this as soon as I can find my razor, you don't have interphase, right? You have prophase, metaphase, telophase. Now, what was the big thing that happened in metaphase? Remember, the chromosomes doubled. Can you remember? The chromosomes doubled. So instead of having 2, you had 4, one of each. Can you remember? Okay. Now in this case, it doesn't double. And if it doesn't double and we pull it apart, just think what's happened. Okay. So we're going to end off. Here we go. If you have a look at it, we are going to end off over here with the 2 cells that we ended off on the last one. Remember, there they are. The 2 cells that we ended off on the last one. And guess what happens? They line up on the equator again. Okay, now if they line up on the equator, remember we started off with the cells that did that and a little piece. There it is. Okay, we'll get to this. This is metaphase. Remember, you get prophase, metaphase, anaphase, and telophase. Okay, so in actual fact, let's start it over here. They still do the same thing. They go line up on the equator. See, they start moving on the equator. The equator is the middle part of the cell. They go and line up on the equator. That's the first thing you must remember. Second thing you must remember is they line up randomly again. Okay, so it doesn't matter if the red is this side or the red is this side. If I'm talking about, remember we had those things where they swapped around. They can line up any way they want. And if they do that, you get random assortment. Can you remember that? Okay. Another thing that happens is, remember, the nucleus divide, the nuclear envelope or the nuclear membrane starts disappearing. It happens in both. Okay? Starts to dissipate or disappear so that we can get hold of it. The centrioles move to the poles, same as mitosis and meiosis 1, right? And the spindle fiber starts forming, as you can see over here. Okay, now, if I go back to what I was saying in the beginning, there it is. Well, we need the purple. Remember, this one goes here and that one goes there. Can you remember that? Okay, and then we had another one where it had that piece there and a little piece over there. Can you see? And then the red. You see what I've done? This is still those 2 that I was talking about. Can you remember? Okay, now when it comes to this part, they didn't double. Can you see there's no doubling at all? Okay, and then you're going to get these spindle fibers. Remember, they're coming across here, the spindle fibers they were coming across. Okay, and you get the same centrioles that are over there. And when they pull, okay, you're going to end off with something that looks like this. You're going to have a chromosome that is red over there. Can you see it? Okay, and you're going to have another one that is purple over there. Can you see it? Okay, then you're going to have another one that is going to be a little bit of red and a little bit of purple. I always use colors for the, oh, come on, purple. A little bit of purple over here, a little bit of purple over there, and you're going to get one that's purple with a little bit of red over here and a little bit of red over there. Can you see it? Now, if this one goes to one cell, and that one goes to another cell, and that one goes to another cell, and that one goes to another cell, what do we have? Does this one look the same as this one? No, they look completely different. does this one look the same as this one? No, it doesn't. What about the top one? This one, does it look the same as this one? No. And does this one look the same as this one? No. They are all different. Can you see how they're all different? Okay. Now, remember, what I've done here is I started off in the other one with 2. Okay. So there was 2 of them. Started off with 2 of them. Okay. Then what happens? We ended off with having 1 in each cell. Come on, not today. We're going to have a cell over here and a cell over here. Okay. That's 2 cells. And then these 2 made that cell, and that one made these 2 cells, and that one made those 2 cells. And they get pulled apart, as you can see in anaphase. Can you see in the anaphase? Okay, which is over here. They get pulled apart, and as they get pulled apart, if you have a look here, very important. Go back down. See, they get pulled apart, and then they split down the middle to create 4 different cells. Even though the colors of them inside here are the same, it doesn't mean that it's what they represent. Okay? And remember, let's say, for example, now this is an example. We started off with this chromosome. Think about it carefully. We had a chromosome where we had your mom, right? And we had your dad, and I'm going to put blue here. Your mom and your dad's chromosomes. Okay. Now, we have 2 of them. We can end up with a chromosome that is identical to your dad. We can end off with a chromosome that's identical to your mom. We can end up with a one where it's got a little bit of your dad and a little bit of your mom up here. And we can end off with one that has got a little bit of your dad down here and a little bit of your mom up here. Now remember, every place on the chromosome represents a different thing. Like this part here, if I put it in red, this part here could be eye color. This part here could be hair color. This part here could be skin color. This part here can be hand size. Not really, but do you understand where I'm coming from? Every little part of that chromosome. So if the black over here is hair color, then the blue over here is hair color. Can you understand how it's working? Okay, so everyone is different. So this might have a complete black one, for instance. This one would be a complete blue one. This one will be a little bit of blue with black at the bottom, and this one will have a bit of black on the top. Come on, with a little blue at the bottom. Can you understand how each one is different? And that means that everything in that cell, that this one will never look identical to this one, or ever look identical to this one or this one. They will always look separate, because each one of those will make a sperm cell or an egg cell. Can you understand how it works? Now that's what mitosis is, and this is meiosis 2. So if we have a look at it from the top again, prophase 2, they line up on the equator. Metaphase 2, they start getting pulled apart slowly, but so they're on the equator, they start getting pulled apart. With anaphase, they get pulled apart quite a bit. With telophase, karyokinesis and cytokinesis takes place. And with cytokinesis, you're going to end up with those 4 cells. Okay? Don't forget, you get 2 types of kinesis. You get karyokinesis, or the splitting of the cytoplasm, and cytokinesis, which is a splitting of the karyokinesis, splitting of the nucleus, cytokinesis, splitting of the cytoplasm. Right, so now that you've got all that and you up to speed and you understand what's going on, relax, let's go have a quick break, and when you get back, we'll do some questions. Are you ready? I've given you all these things together, right? I've got the meiosis 1, I've got meiosis 2, and you do understand them, right? Make sure you concentrate. We're going to go into the difference between them, okay? But, the difference between the process 1 and process 2. So, here we go. First question, okay? Here we go. Briefly, and I'm going to highlight it in pink, okay? Briefly describe the difference between prophase 1 and 2. Okay? Prophase 1 and 2. And how many cells are found at the end of prophase 2? Carefully. Okay? I'm going to give you 2 minutes. I want you to think about it. because you need to understand exactly what's going on to be able to do these questions. Are you ready? Go. Okay, so you have looked at these questions and you have given a lot of thought to them, right? Let's see what we can get out of them. First thing, if we get a bit of pink going there again today. Now, you know what? Let's go blue. We had pink in the last one. Here we go. Briefly describe the difference between prophase 1 and prophase 2. Okay, let's see. In prophase 1, crossing over occurs and the spindle fibers form. Okay? In prophase 2, no crossing over occurs and spindle fibers reform and attach to the centrosomes. Now, what does that actually mean? Okay, remember when we had a look at prophase 1, we had where they crossed over and you had 2 that did that. Can you remember? And we got the place where over here was the chiasma, okay, or chiasmata if it's more than one, okay. And then you got to the spindle fibers were starting to form. Remember, they came from the centrioles. Spindle fibers started to form, and they wanted to pull those apart, but they didn't pull them apart yet. When it came to prophase 2, what happened? Prophase 2, they didn't cross over. They just sat there on the line, one over there and one over there. And what happens is that one pulled that way, that one pulled that way, that one pulled that way, that one pulled that way. Can you understand how that worked? Okay, this one, this whole piece. Well, let me put it in black. So let me undo that. No, man. Come on. Today we're not working together. Okay? You know what it is? It's me. It can only be me. Right, let's have a look. If I have a look at this, that one there, okay, is going to be pulled in that direction. And this one here will be pulled in that direction. Okay? Where here, half of it gets pulled that way, and here, half of it gets pulled that way. Can you understand how it works? Okay? And that's the difference between prophase 2 and prophase 1. A little question for you while you sitting back there and you're thinking about it. What is the difference between prophase and prophase 1? Think about that one. What is the difference between prophase and prophase 1? It's very simple. Okay? Think about it. In prophase and prophase 1. In prophase, when we get, I'm just going to see if I can lengthen this up a little bit. If I can, let's move it down. There we go. Give you a little bit of space there. Okay? Watch out. In prophase 1, just prophase, okay, you're going to have them lining up next to each other here, okay, and they're going to be pulled that way to the centrioles. You happy with that? In prophase 1, they're going to be lined up, almost the same. The only difference is now they're going through that process called crossing over, and then they're going to be pulled to the side. Can you see the difference? Okay, remember, look at the crossing over. This here is prophase 1, and this is just prophase. This is mitosis, and this one is meiosis.